Now we are going to discuss some questions which are frequently asked in the spotting exams. Like, take for example this question. Okay, identify the procedure being done over here. What is A and B? Okay, the same question can also be framed in identify the procedure being displayed. Okay, what is the physiological significance of the procedure in the nerve muscle preparation? So both of this procedure is showing you and you giving you in experimental is done in experimental physiology to give you nerve muscle preparation. So basically it's a preparation of a nerve and a muscle. Okay, so we isolate the gastrocnemius muscle of an animal along with the nerve, usually the sciatic nerve. Okay, so this combination of these two structures is called as nerve muscle preparation. Now before getting or isolating these structures, we have to do some procedures. The procedures are A and B. Okay, so A is denoting over here stunning step of the nerve muscle preparation, and B is showing you over here a pitting step, P-I-T-H-I-H, pitting step. Okay, what is the meaning of the word stunning? It's literally, it means stunned the animal. Okay, we grab hold of the hind limb of the animal and with a force we strike the forehead of the animal on a hard surface. Okay, that will instantaneously make the animal go stunned. Okay, so obviously unconscious animal and the vitals would be intact. Okay, but if you are giving some jerks, electrical shocks, the animal might react. Okay, there will be some reflexes which will be hindering our experiment. So merely doing this turning step is not sufficient for our isolation. Okay, next step, what we do the pitting step. In the pitting, what happens, we use a specialized needle called as pitting needle. Okay, so here the needle is being shown over here. Now we take a stunned frog, okay, from A, and what we do, we grab hold of the anterior part of the head and flex it with our one hand. Okay, in such a manner the head is completely flexed and touching the dorsal part of the ventral part of the body. So what will happen, triangular shaped depression is visible, okay, somewhere around the midline of the head. Okay, so if you are tracing down the tympanic membrane of both sides, okay, so both sides tympanic membrane, you draw a imaginary line, the line which is crossing the midline of the body, that is the point at which you should get a triangular depression area. Okay, so we isolate that area and we insert the pitting needle. Okay, it's a very sharp long needle. We insert this needle with the other hand and we first of all go forward, okay, and try to destroy the whole brain, and then we go backward, we try to destroy the whole spinal cord of the animal. Okay, we give some twist, okay, we give some twist and turn and we completely destroy the brain and spinal cord. This is the step. Okay, so the stunning of the animal is A and B is pitting of the animal. Now why do we do this in the nerve muscle preparation is the physiological significance of this procedure means it's an human way, of killing the animal for experiment. Okay, so whatever experiment we are going to do, so it's merely cutting the head of the animal, and it is not sufficient enough for doing this experiment. It looks very bad, so this is one way to interpret. So physiologically what happens, by doing this method, what happens, we are actually destroying the motor neurons, okay, that is present in the spinal cord, okay, which if was not destroyed would have led to some reflex withdrawal. Okay, so if we are doing an experiment on the muscle or the nerve, okay, supposedly you are taking this nerve and you are stimulating with an electrical shock. If we did not do pitting step, what will happen, there will be withdrawal response of the leg and the animal will react. Okay, so and third is it confirms the death of the animal. Okay, so we are absolutely sure after doing this turning and pitting step that the animal won't respond during the experiment. Okay, now moving on to our next experiment. Like, questions might be asked in this scenario, like identify the curve displayed. Okay, what interval is shown in the level A and B. Okay, so this diagram that is showing you is a typical simple muscle curve. Okay, it's a curve which is generated when we are giving electrical shock with the help of an electrode on a nerve muscle preparation. The muscle will contract, and the muscle is tied with a lever. Okay, and the lever is connected in turn to the moving chymogram, okay, which depicts a curve. Okay, that curve we are getting over here. So this is, if the answer is identify a curve, just mere writing simple muscle curve is sufficient enough. Okay, sometimes this is also called as simple muscle twitch. Okay, a momentary movement or contraction of a muscle is also called as twitch. So sometimes we call it this also. Okay, what interval is shown in A and B? So this is the interval A and this is the interval B. Now typically when we get a simple muscle curve, there are some points that you should remember. Okay, so a curve is always drawn on a baseline, okay, over which we determine the curve's duration as well as amplitude. Okay, and below that we have some sinusoidal waves. These are nothing but the time frame or the time tracing. Okay, now at the beginning of the experiment we get a line over here. This line is showing you the PS point. Okay, so this is point of stimulation. Okay, then from PS we get another interval which is called as LP. Okay, so if this is the curve, this is the point of stimulation at which the response is generated due to the electrical shock, and it takes momentary some time to show the response on the curve, and as soon as it goes away from the baseline till it reaches peak, okay, this particular point is called as point of contraction. Okay, from this point the contraction starts and till it reaches the peak. Okay, so this, from this point to this point is called as the contraction period. Okay, and from the peak till it reaches the baseline is called as the relaxation period. Okay, and the interval from the point of stimulation to the point of contact is called as LP or latent period. Latent period. Okay, so latent period has some physiological significance also, which I'm going to tell you. So in this answering this question, identify curve, simple muscle curve is a simple muscle twitch also. What interval is shown in A and B? A is showing you the contraction period and B is showing you the relaxation period. Now immediately when the curve touches the baseline, it is not stopping, it is showing you some curvilinear curves. This is called as physiological curve. Now it is because of the inertia of movement of the metallic instrument, that is the stylus or the lever. So it keeps on moving till it goes into rest. Okay, so now you should also know about the duration of this periods. The latent period is approximately 0.01 seconds, or you can also write it as 10 milliseconds. Contraction period, the time period is around 0.04 seconds, or it's sometimes written as 20 to 40 millisecond. And relaxation period, the time period is 0.5 second, or sometimes written as 50 millisecond. Okay, so the total duration, if you can see, is total duration of twitch will be 0.1 second. Okay, now apart from this, questions are also asked related to this latent period. Okay, this latent period is very important. Sometimes questions also come, what is the physiological significance of latent period? Latent period, physiological significance. Okay, so number one point is, it is the time taken for the stimulus to travel along nerve, okay, to the neuromuscular junction. Okay, the time it takes to travel, if supposedly you are giving the response in the nerve, okay, and the nerve takes some time to go from here to the neuromuscular junction, and the response is generated by the contraction of the muscle. So suppose this is the muscle and this is the nerve, you are giving stimulation over here, so it takes time. Okay, so this is the time period taken for traveling. Okay, it is also can be written as time taken for impulse to cross the neuromuscular junction to stimulate the muscle under experiment. Okay, the same thing only. You can also write, it is the time taken for the excitation, contraction, coupling to occur. You know, muscle contracts because of excitation, contraction, coupling reaction. Okay, so it is also the time it needs to do this reaction. Okay, it can also be written as the time taken by the lever to overcome inertia, okay, of rest. And the fifth point is, it's also can be written as the time taken to overcome the viscous resistance of the muscle. Okay, next question for our discussion in spotting is, take for example this question: identify the curve displayed, write the physiological significance. Okay, anyways, it's all written over here. Sometimes the examiner or the question that will be given, they might hide you this part also. Okay, they might give you one particular number also, or sometimes it could be visible. Okay, take for example this one. Okay, so identify the chart displayed, name the part marks X. Okay, so this is a chart which is showing you the variation effects of temperature, effects of temperature on simple muscle curve or simple muscle twitch. Okay, so you can see when the temperature is more, okay, 38 degrees Celsius, that is high temperature, you can see the amplitude of the curve is more. Okay, as well as the latent period is less. But when you are taking the curve on a temperature that is 16 degree or 4 degrees Celsius, so you can see the amplitude of the curve is less. Okay, as well as the duration, the latent period increases. So these are the two curves, and the middle curve is a simple muscle twitch which is done at a room temperature, that is 27 degree, around 27 degrees Celsius. So when we are getting this curve at a 38 degrees Celsius Ringer solution, okay, by the way, Ringer solutions are used in experimental physiology. Whatever tissues are you're using, you should always use a Ringer solution which has an isotonic concentration with that of the extracellular fluid of the tissues in the amphibians. So what happens, the tissues are viable for few more minutes for our experiment to be completed. Anyways, so if we are using a warmer Ringer solution, or we are using a warm solution for our experiment, what happens to the simple muscle twitch, there will be increase in the latent in the amplitude of the curve. Okay, in the amplitude of curve there will be decrease in the latent period of the curve. Okay, and the relaxation period will also decrease. Okay, now why these phenomena are occurring? The reasons for these are increase in conduction velocity in the nerve. Okay, when the temperature is higher, relative conduction velocity in the nerve increases. Okay, similarly there will be increase in rate of neuromuscular transmission. Okay, that means the transmission between the nerve and the muscles, that is the neuromuscular junction, the chemicals will be secreted at a much easier way, or the viscosity decreases, so there will be increase in transmission. Also, inertia of lever is overcome faster at a higher temperature, overcome faster. Okay, now these questions usually come on a physiological basis. Now physiological significance of these effects are, the contraction period is decreasing. Okay, the relaxation period is decreasing. Okay, the latent period is decreasing, but the overall amplitude is increasing. Why? The reason is, number one reason or physiological reason is due to activation of myosin ATPase activity. Okay, so this enzyme's activity will increase. Also, there will be decrease resistance of the muscle to contractions as viscosity decreases. And third physiological significance is that the amplitude that we are getting to be higher in higher temperature is because of amplitude contraction increases because there is increased enzymatic and chemical activities in the muscle. Okay, so these are the physiological significance that is taking place in higher temperature. In the cold temperature, that is cold ringer temperature, that is at 4 degrees Celsius to 16 degrees Celsius ringer solution, the opposite effect occurs. Okay, there will be decrease in activation of the myosin ATPase activity, and there will be increased resistance of the muscles to contractions because there will be more viscosity, and an amplitude will decrease because there will be decrease in the amplitudes decrease in the enzymatic and chemical activities in the muscle. So next question for our discussion is identify the effects observed in curve 3 and 5. Okay, so these curves are a simple muscle curve with some variations in the morphology. Next question, this question can also be framed like identify the chart. Okay, write the physiological significance of the chart. They might give you just a single curve or might give you a two curve. Okay, or they might give you the whole procedure of the experiment. Okay, and they should mark it with 1 to 6 curves, and they give you the random number of curve and identify what is happening in 6. Okay, or identify the 6 curve or 4 curve, whatever curve. Okay, write the physiological significance of the curve. Okay, so anyways, in the previous curves we saw that if we are giving a simple muscle twitch or there is a simple one single stimulus, that will lead to formation of a single curve that is called a simple muscle curve. Okay, being this is the point of stimulation P1. Okay, now it has a point of stimulation, it has a latent period. Okay, so now this is a latent period. We know from the point of contraction to the peak apex of the contraction, that's called as the contraction period. This is a relaxation period. Okay, so as soon as it touches the baseline, now what will happen if the chymogram is rotated 360 degree and at the same nerve muscle preparation point we are stimulating another curve, another stimulation, and we name that stimulation to be P2. Okay, so based upon the point of contact of the second shock, the different curves that we get, it depends upon the position or the timing of the second stimulation. If the second stimulation is falling in such a way, it is falling at the latent period, that you will get different curve. If it is falling on the contraction period, it will get a different curve, and reduction period you will get another curve, and at the end of the reduction period you get another curve. Take for example this curve only. Okay, curve 1. Okay, this is the curve, this is the basic curve with which we are starting. So this is the whole experiment, by the way, is called as effect of, let me write you first of all, identify the curve. The curve is effect of two successive stimuli on simple muscle curve or simple muscle twitch. Okay, now the first curve is a simple muscle curve, okay, with having a one stimulation. Okay, the single stimulation is giving you a simple muscle twitch. Now in this second curve you can see there is P1 and there is P2. Now in this curve specifically you can see the P2 is falling if supposedly this is the latent period. Okay, now immediately when the P1 was falling, okay, just within few milliseconds another P stimulation is coming. Okay, so that means the two shocks are coming very close. Okay, so the time frame between the first and the second is very close. Okay, so in this case you can see almost it's equal. Okay, but on the other hand, if P2 is falling a little bit farther away, okay, as compared to that of the P1, okay, you get the amplitude to be a little bit higher than this first curve. Okay, similarly, if supposedly the P1 is given at this particular point, that is this point, and it has gone or traveled at least 0.1 or 1.2 seconds, that means it has crossed the latent period, and then you are getting the P2, then you get another curve which has a even bigger height or amplitude. Okay, it has a lesser total duration of the twitch. Okay, so you will get a typical feature of this curve. Okay, now the 5 and 6 are kind of an atypical curve. Okay, so it does not look like a bell-shaped curve or your total sigmoid curve. See, if this is the P1, it has crossed the latent period, the contraction period has completed. Okay, it is touching your this peak. Okay, now what happens, just when it is relaxing, okay, just when it is 1/3 of the relaxation period is covered, a P2 simulation comes, that is the second simulation. Now immediately it responds with another curve which is a bigger curve as compared to the first curve. So you get a typical feature of two peaks in this curve which are fused at the middle, whereas take a note on the sixth curve, this is the P1 curve. Okay, the latent period is gone, so because of the P curve we get a contraction period and also we get a relaxation period. Okay, and you see over here the curve is touching the baseline. Okay, almost is touching the baseline. Now after it touching the baseline we are getting a second stimulation, then also we are getting a two peak. Okay, so here also we are getting two peaks, but these two curves are not fusing to each other. Okay, that's the difference between the 5 and the 6 curve. Anyways, all these curves are showing you the effects of two successive stimuli on the simple muscle curve. You are doing the experiment of a skeletal muscle and you are getting this curve. So if the question is framed like identify the curve which is mentioned in curve 5 supposedly, what you are going to say that this answer to this question will be the fifth curve is showing you the effect of two successive stimuli on the simple muscle curve with second stimuli falling on the relaxation period of first stimuli. Okay, that's all, that's it. Now the physiological reasoning. Okay, what is the physiological significance of the curve? Now this physiological significance it signifies that the mechanical response of a electrical stimulation is that the simple muscle curve is responsive to the second stimulation anywhere around the simple muscle curve, anywhere around the simple muscle curve, except except the except the first half of the latent period. That's why you have seen that this curve, the second stimulation was falling at the first half of the latent period, so almost the height of the curve was same only, so it was not responsive. So the second curve is showing you the effect of this same sentence, effect of two successive stimuli, where the second stimuli is falling on the first half of latent period of first stimulation. That's it. So that's the naming of the curve. Okay, the reason for this physiological significance of the increased amplitude as well as decrease in the latent period, okay, as well as decreasing in the total contraction period is what happens. There will be the beneficial effect. Okay, there will be a beneficial effect of the first curve. Okay, you have to know what is the beneficial effect. So the physiological significance of all these curves are because of the beneficial effect of the first curve. Okay, so beneficial effect are nothing but all those factors which are needed for your excitation contraction coupling. That is, it happens because of the presence of number 1 point, calcium ions, which are needed for excitation contraction coupling, which was released because for the first curve, that is the P1 curve, it's remaining in the vicinity. So when the second stimulation arrives, it is already present. Okay, so some of the after the simple muscle twitch is completed of the P1 curve, they will revert back to the terminal cistern. Okay, but since it is a very closely spaced experiment of second successive stimuli, some calcium will be left, which will be easy for the next curve to show the response, and that's why you are getting a bigger response. Okay, so that's why you are getting a bigger response of the curve. Also, what happens because of the first curve, what happens, there will be increase in the temperature. Okay, there will be decrease in viscosity. Okay, all these factors will lead to increase in the amplitude of the second curve, provided you are giving the second stimulation after the first half of the latent period, because first half of the latent period is the first half of latent period are absolutely refractory period, so no response will be generated. Okay, so next question for our discussion is identify the chart, write the physiological significance. Okay, so this is a curve which is showing you the conduction velocity of nerve impulse in a frog, sciatic nerve. So here it's a simple muscle curve only, but having two simple muscle curves with different nomenclatures are there. Okay, M curve and V curve. So this M curves is signifying you the simple muscle curve when we are getting this simple muscle twitch when we are stimulating the nerve at the muscular end, and V is showing you the curve when we are stimulating the nerve at the vertebral end. Now for this you should remember what is the nerve muscle preparation. You remember from the first question what is the nerve muscle preparation. I told you we isolate from a frog a gastrocnemius muscle. Okay, attached to a sciatic nerve, okay, which is attached to a vertebra. Okay, the sacral vertebrae are there, S2 3, okay, S1 2 3. So this experiment is conducted in two phases. In the first phase, what we do, we stimulate the nerve somewhere around here. The nerve will stimulate, the transmission of the nerve impulse will pass from this nerve end, and it will reach the muscular end, and the muscle will contract, okay, and it will pull the thread of the lever, and the chymogram depiction of the simple muscle will be shown. Okay, supposedly this is the M curve. Okay, so let us denote this as, sorry, so since it's near to the vertebra, so this is near to the vertebra, let's denote this as a V. Okay, so this is the V curve. Okay, and this is supposedly the point of stimulation for the V curve. Now the same point only, supposedly same numbers of patient, we are giving the stimulation this time around near to the muscular end. Okay, you are stimulating the nerve near to the muscular end. Since it's near the muscle, we are denoting the curve to be M. Now the nerve takes little shorter time as compared to the V curve, and it stimulates the nerve muscle, and we get another curve. Okay, now this time around we will see that the curve is showing you a little bit lesser latent period, almost the same height, but the duration will be decreased. Okay, now why it is happening? Since the muscular end is nearer to that of the vertebral end, it is taking lesser amount of time for traveling to of the nerve signal, as well as the beneficial effects that are generated, it needs shorter time frame. Okay, the ATPase activity, the enzymatic activities, as well as myosin head and actin filament cross bridging, and the injection of the vesicle neurotransmitter from the vesicles, it takes lesser amount of time for generation of the impulse. Okay, so we are getting these two curves. Now these two curves, the distinguishing point is there will be lesser latent period is compared to in the muscle curve as compared to that of the vertebral curve. Okay, the next curve for our discussion is identify the curve displayed, write its physiological significance. Okay, so you can see in this curve there is three curves are shown. Okay, all these curves are simple muscle curve only, but having three simple muscle curves. Okay, some letters are written like 1G, 2G, then no load, sometimes SMC is written. Okay, so like this the exact time frame could be the legends could be written. Okay, here also another curve with having different morphology of the curve, like the no load is written at the top, and here the no load is written at the very middle. Okay, so these two curves are the same curve only. Okay, so basically this curve is showing you, the topmost curve is showing you, it's a curve of effect of load on simple muscle curve, and this curve is a pre-load curve. Okay, these two curves are the effects of load on simple muscle curve, and it is showing you the after load, after LOAD, load SMC curve. Okay, so simple way to memorize is that the simple muscle curve or the no load curve. Okay, so this experiment is conducted is by attaching the load to a muscle even before the stimulation is given to the nerve muscle preparation. Okay, if supposedly you are loading the muscle and priming the muscle with a counterpoise weight, so that you are slightly stretching the muscle even before you are giving stimulation to the nerve muscle preparation. So what will happen, there will be a beneficial effect of this priming of the muscle, and you will get the curves to have next curve. When we are going to get another curve with attaching an added weight, you will get a increased efficiency of the muscle, okay, because of the priming of the muscle. By slightly stretching before the experiment, you are getting a higher amplitude curve, okay, with lesser amplitude, less latent period, and higher amplitude curve and lesser duration. But since it's a very small tissue, so it will have a physiological limit, after which if you are adding another gram, what will happen, this all the myosin actin cross bridging will be overlapped, okay, so it will not have an optimal cross bridging, okay. And what will happen, the next gram that we add, even after you have primed the muscle with a no load curve with a counterpoise weight, you will get a detrimental effect of the next gram that is added. That's a no load for you. So that's a preload experiment for you. In an afterload, what happens, the weights or the counterpoise weight you are adding to the nerve muscle preparation are acting only when the stimulation or you are giving the shock to the muscle, okay, or the nerve muscle preparation, okay. So the simple muscle curve that we are getting in a no load state, that is when the weights are not acting, or not even the counterpoise weights are acting on the muscle, what will happen, you will get a simple muscle curve. But after adding the next grams, what happened, it has already crossed the physiological limit of its working efficiency, okay. So the counterpoise weight will be acting over that, the 1 gram also is acting simultaneously in the next step of the experiment. This is a 1 gram experiment. So this time around, what will happen, it will have a detrimental effect, and it will have a lesser amplitude and longer latent period. Now obviously the next gram you add, it will have a more detrimental effect, so you will get a more latent period and a lesser amplitude, and you will get a curve like this, okay. So it's a very easy curve to understand. So if you're getting a no load curve at the middle, it's a preload curve. If you're getting no load curve at the very top, it's a afterload curve. What is the physiological limit? Physiological limit is priming of the muscle with a counterpoise weight before the experiment is started. It gives you the preload phenomenon, and you will have beneficial effect of loading the curve, loading the muscle with the weight. Whereas in case of the afterload, what will happen, since it's not primed, the muscle is not under effect of slight stretching of the elastic component of the muscle. So even after adding the 1 gram, immediate 1 gram, it will cross the physiological limit of its efficiency, working efficiency, and will have a detrimental effect of the working. So next question for our discussion is, identify the curve mentioned X. What is MTF? And also the question can be framed that identify A, okay, or the curve A, or they might give you B curve, C curve, or D curve. You have to know all the other curves also, and what are these curves as a whole. So to answer this, the curve that we can see in both of these questions is showing you the genesis of tetanus, okay. So this is curve which is showing you genesis of tetanus, and what is the tetanus, then that you are going to know, okay. So the definition of tetanus is, tetanus refers to a state of sustained contraction of muscle without relaxation without relaxation due to repeated stimulation. Okay, so mind that this experiment is conducted with a specialized instrument called as either Du Bois inductorium, or we use a vibrating reed with interrupter. Okay, with interrupter. Okay, so we might use these two instruments, Du Bois inductorium or vibrating reed, to do this experiment, because we have to give repeated stimulation in this experiment. Mind that this experiment is achievable only when we are using a higher frequency, okay, of stimulation. That means the stimulation has to be coming at least at a rate of 15 to 20 times per second. Okay, if you are getting the frequency of stimulation or lesser than that, you might not be able to achieve that tetanus. Okay, by the way, in whole this experiment, the tetanus is only this part only. Okay, only the D curve is showing you tetanus. Okay, the rest are the curves which are not showing you tetanus, because in tetanus there has to be sustained contraction. Okay, and by the way, this curve is showing you the simple muscle curve only. Okay, that we learned in the few last questions, that if you give a stimulation, from the point of stimulation we get a latent period, then contraction and relaxation period. That was happening with a one particular single shock. Okay, if supposedly you are rotating the kymogram at a very slow speed, okay, and you are giving repeated stimulation, what will happen? This point of stimulation will be like that. Okay, there will be small latent period and you will get a contraction period, then you will get a relaxation period. It will cross the baseline, then it is relaxed, then again another stimulation curve P2, like this you get another curve. Okay, like this you get another curve. So every time when it touches the baseline, you get a curve called as A. So this A curve is given a special name called as triple curve. Okay, or staircase curve or step ladder curve, whatever you want to write, you can write. Okay, it looks like a step, okay, gradually increasing in height. So that's a typical feature. So you will have a curve having spikes which are touching the baseline every time. Okay, touching the baseline, this is the main point to know, distinguish between the A, B and C. Okay, it's touching the baseline. Now why this triple curve is occurring? Because of the beneficial effect of the first curve. I told you, in the effect of two successive stimuli, what happens when the first curve was formed? Because of that, the calciums are needed, the temperature has to be increased. Okay, all the myosin and actin heads has to be forming a cross bridge. Okay, all those beneficial effects of nutrients and neurotransmitters that are needed for the muscle contraction, that has to be present. Now supposedly first curve is showing you this curve. Now at this particular point, the calciums are reverting back to the terminal systems of the sarcoplasmic reticulum, but some of the calcium will be still left at this particular point. So because of that beneficial effect, the other curve will get a bigger amplitude curve. Okay, like this it keep on happening till it reaches a physiological limit of optimal limit. Okay, then it becomes stagnant. Okay, till you stop the experiment of stimulation. Okay, so that's called the triple curve. This is the triple for you. Now what is this B? Now in the B you can see the curve is somewhat like this. You give a stimulation, point of stimulation, you get a latent period, you get a spike. Okay, contraction period, you get a relaxation period. Okay, supposedly before completion of the latent period. Okay, this is latent period, contraction period, this is sorry, lateral period, this is contraction period. Okay, now when it's coming down, supposedly at this particular point only, when it's coming down at the relaxation phase, it is encountering another stimulation P2. Okay, then because of the beneficial effect, it will have another curve. Now again it will encounter another stimulation P2, P3. So like this it will keep on happening. Okay, we will get as individual spikes which are not touching the baseline. This curve or the B curve is called as incomplete tetanus. Okay, the same thing happens with the C curve also. Okay, the C curve is happening with some smooth sustained contraction at the end. Okay, now A curve is occurring when you're giving a stimulation at the rate of 5 to 10 hertz. Okay, B curve is occurring if you're giving a stimulation at the rate of 15 to 20 hertz. Okay, C curve is occurring if you are giving a stimulation of around 30 hertz. Okay, and tetanus happens usually in a frog's muscle when you're giving a stimulation at the rate of 40 hertz. Okay, now this A curve gives given a special name called as strip A. The C curve is again given a special name called as the clonus, and D curve is nothing but the hero of the experiment, is the tetanus. Okay, so the next question is, what is MTF? What is minimal tetanizing frequency? Now I told you every muscle. Now this tetanus is achieved when we are giving a higher frequency of stimulation, that is the repeated stimulation has to be occurring. Okay, so when does this happen? It will form a tetanus. Now it will depend upon the length of the muscle, the diameter of the muscle. As big the muscle becomes, it will have more amount of calcium, so it will have more amount of time for showing you the tetanus. Okay, you might get D, E, F, G, H, like so many curves we will get. Okay, or it might be that if you are increasing the temperature, you might tend to get lesser amount of more amount of curves. Okay, so the factors of MTF and the time frame, the factors are many. We will discuss one by one. So first of all, the definition of minimal tetanizing frequency. Minimal tetanizing frequency. Minimal tetanizing frequency. Now that's a frequency for you. Okay, so this minimal tetanizing frequency of a muscle can be defined as, or can be calculated from the simple muscle curve by doing a formula of MTF is equals to 1 by contraction period. Okay, so supposedly you are getting a latent period, contraction period, and contraction phase, relaxation phase. If you somehow can measure this particular duration, then you can get the MTF. Okay, so basically MTF is a frequency at which we get the muscle tetanized. Okay, and you then write the formula. Okay, so usually MTF in a frog's muscle is, we know contraction period is 0.04 seconds, so we get a frequency of 25 hertz. So that's the normal standard MTF. Now the factors which affect the MTF are, okay, first is the type of the muscle. Okay, so usually in a slow muscle the MTF is low because you need lesser amount of frequency to achieve the tetanus. So what happens in a slower muscle, the muscle will be in a slow sustained contraction, so there will be a longer contraction period. Okay, the contraction phase will be longer, so we might need a lesser amount of stimulation, lesser frequency of stimulation for achieving this. Whereas in case of a fast stimulating muscle where the contraction phase is lesser, so we might need a higher frequency. Okay, the subsequent stimulation should come or arrive at a very quick interval. Okay, example of one of those is slow muscle is like soleus. Okay, the calf muscles. Okay, if you take the calf muscles, and we are doing the experiment of the frog on a gastrocnemius muscle, so it's a calf muscle only. So in those muscle we need a frequency which is less, relatively lesser. If you take supposedly the ocular muscles, okay, extraocular muscles, okay, where the contraction phase are very less, so in those case we might need a higher frequency, titillizing frequency. Okay, second factor which affects is the temperature. Okay, the temperature, as you know, temperature helps in assisting the transmission of the conduction. Okay, so a hotter temperature is very viable to achieve your tetanus. Okay, if you are increasing the temperature, you will tend to get a higher frequency of so sorry MTF if inversely related to temperature. That means if supposedly you are increasing the temperature, what will happen, there will be lesser amount of viscosity. Okay, the transmission will be very easy, so you will need higher frequency rate. Okay, so that means the muscles, the next subsequent contraction should be coming very close to each other. Whereas if you're doing this experiment on a cold setup, okay, cold setup, then the contraction phases will be spaced out. Okay, it will be slower. Okay, the viscosity will be increasing and it will be having a sluggish contraction period, so you might need a lesser frequency. Okay, the third scenario for your factor is load. As I told you, load helps in priming the muscle. Okay, so formula wise, MTF is inversely related to load. If supposedly if you are adding a load, okay, increasing the load, you will tend to get a lesser frequency of MTF. Okay, suppose and if you are decreasing the load, the MTF has to be seen, it increases. So next question for our discussion is identify the curve, what is X and Y. Okay, or identify a curve, what is A and B. Okay, now this curve is showing you the normal cardiogram of frog's heart. Okay, so typically when you are doing the experiment of instead of the nerve muscle preparation, you are using the heart muscle or cardiac muscle, you are getting a curve like this. Okay, so in this curve, take for example this curve. Okay, so this curve has one arm which is straight without having any angulations, whereas this curve has two angulations. Okay, on the other hand, the below curve is showing you a single notch on the downstroke, so this is called as the upstroke curve and this is called as the downstroke. So this is called as the upstroke curve and this is called as the downstroke curve. Okay, so upstroke curve is a very smooth curve, whereas as compared to that, the downstroke curve is a curved or having two notches. Okay, in the below picture you can see only a single notch is there. Now this part or the upstroke part is called as your diastolic phase. Diastolic phase. Okay, so down upstroke it shows the diastolic phase of heart, whereas upstroke is divided into basically three parts. Okay, the first part, the X is showing you systolic phase of sinus venosus as compared to that of the human heart, whose pacemaker cell is the sinoatrial node. In case of the frog is the sinus venosus, which does the work of the sinoatrial node. Okay, and the systolic phase of that tissue is shown by X, whereas A is called as, next is A is showing you the systolic phase of atria, whereas the Y is showing you the systolic phase of ventricle. Now you might ask, where is the diastolic, atria, sinus venosus and ventricle phase? Now all this combined, the sinus venosus, atria and ventricle together, are the events are shown in this, the upstroke curve. Okay, so X is showing you the sinus venosus systolic phase and Y is showing you. Now sometimes this curve is shown as S curve, this is shown as A curve, and this is shown as V curve. Now in this, A is showing you what? The downstroke. Okay, so the downstroke, it is showing you the downstroke, so it has to be the systolic phase of the ventricle, systolic phase of ventricles. Okay, now B is showing you what? B is actually aligning with this one, so it is showing you the upstroke, so that means it's showing you the diastolic phase, just the diastolic phase of the heart. Okay, so next question is, identify the curves displayed, give the physiological significance of the curve. Okay, the question can also be framed in way of like, identify the curve, what are the effects observed in the curve B. Sometimes in certain books and some people like to answer in this way. Okay, so don't get confused with those answers. So this is the question which showing you a curve which looks like a cardiogram, and that's written normal and a worm. Okay, obviously this, from this point to this point, is the point at which the heart tissue or the cardiac tissue has been dipped in a ringer solution with a warm temperature, and in this case it is showing you from this point to this point the cardiac tissue was dipped in a ringer solution having a temperature of 15 degrees Celsius. It is very much similar to that experiment of effect of temperature of simple muscle curve. That was a simple muscle curve, but all the physiological explanation related to that can be explained with this experiment also. As we are increasing the temperature, what will happen? First effect is that we will get the curves to be spacing close together. Okay, also you will get to see that the curves are little bit higher in amplitude. It's not so appreciable over here. So take for example this curve also, so you will get the curves to be increasing in an amplitude. Okay, and we will have a increase in the rate of the curve, heart rate of the, by the way, this is the sinus venosus in which the effect is slightly different. Okay, these are the curves which are found in the other tissue, that is the atria, ventricles. Okay, and in this case the sinus venosus, the pacemaker activity is a little bit different, in which the rate of the heart rate will be different, but as a whole the beneficial effect in case of the cardiac tissue Usually these curves are only given because extracting the sinus venosus and doing the experiment is very difficult, so usually you will get in the exam these kind of a curves only, but you should also know this phenomenon also. Okay, so it's an atypical feature of a sinus venosus. So let's concentrate on this particular part. So when you increase the temperature, what happens? The myosin ATPase activity increases, okay, and it decreases the resistance of the muscle to contraction, okay, and also the viscosity of the muscle. Because of the increased temperature, the viscosity will decrease and you are tend to get a bigger amplitude curve, while in case of your lesser temperature you will have the opposite effect. So let's learn one by one. So what will happen to the sinus venosus? Okay, so when there is a cold temperature, when we are exposing the sinus venosus, that is the pacemaker of the frog, we are adding the cold ringer. What happens? The first effect that we can obviously see is that there will be decrease in the heart rate, okay, and we are going to get increase in the force of contraction. Okay, now in this case, so the heart rate is the primary effect, okay, is the primary effect of the cold ringer solution. It what happens when the cold ringer solution is given? It directly affects the pacemaker cell and it inhibits the pacemaker activity, so as a result of which, what happens? The heart will be pumping at a very slower pace, okay, the heart will be pumping at a very slower pace. Now because of that, what happened? The end diastolic volume will be increasing, okay. Now because of increase in the end diastolic volume, what will happen? The Frank Starling law will come into play. And because of that, we know as the initial length of the muscle increases, the force of contraction increases. Now we get this effect. So the secondary effect is force of contraction increase. So in cold ringer, in sinus venosus, we will get the effect of increasing in force of contraction, which is a secondary effect, the primary effect being the decrease in heart rate. Whereas when we are supplying the warm ringer solution, what happens? The opposite effect occurs, that is, it will directly affect the sinus venosus and will increase the heart rate. Now as you know, when the heart rate is increased, what happens? There will be lesser amount of time for the ventricles to fill the blood, so you will get a lesser end diastolic volume, and because of the Frank Starling law, what will happen? You will get a decrease in the force of contraction. So that is happening in the warm ringer solution, okay. So you're getting lesser amplitude and you're getting increased heart rate, okay. Whereas in case of your ventricles, atria and ventricles, both of those will be showing you the feature that is displayed in the upper curves, okay. Let's talk about the ventricles as a source, okay. Ventricles. Now what will happen to the ventricles is that when we are supplying the cold ringer, what will happen is that the cold ringer will affect the myocardial activity or myocardial contractibility, okay. It will inhibit or decrease the activity of this heart. So as a result, what happens? The rate of pumping will be decreased, okay. So what happens? The force of contraction will be affected and you will get a lesser force of contraction, or the amplitude that you get in a cold ringer. What will happen? It will have lesser amplitude, but the heart rate won't be affected. It will neither decrease or neither increase, because the heart rate is regulated by sinus venosus. So that's why in the warm solution you can see almost it's equal, okay. Almost the rates are equal, okay. So the force of contraction, whatever this cold ringers. Okay, we are discussing about the cold ringer solution. So in the cold ringer you can see the force of contraction, it decreases, okay, as compared to that of the heart rate. It always remains the same. Anyways, the picture is not typically depicted. Okay, the typical depiction is little different. Okay, so forgive that picture. So it should be like that. Whereas in case of your warm lingered lactate solution, what happens, you will get the myocardial activity to be increasing, myocardial activity, contractility to be increasing, and you will get a increased force of contraction. If supposedly this is the normal cardiogram, and you will get a higher amplitude cardiogram with maintaining the rate. Okay, the rate will be same only, but the amplitude will increase. So then the next question is, identify the part marks X in the curve, write the physiological significance of the curve. So this curve is showing you the effect of extrasystole and compensatory pause in cardiogram of frog. Okay, so by this time you know what is a normal cardiogram, having a diastole then a systolic phase. Okay, so this is the normal cardiogram. Now in this normal cardiogram, I told you there is a diastolic phase, there is a systolic phase. Now supposedly in this cardiogram, you know there is a relative refractory period, there is an absolute refractory period. If supposedly you are encountering or you are giving an external stimulation, in this case we are giving an electrical shock to the cardiac tissue. Okay, so in a cardiac phase, in a whole cardiac cycle, this systolic phase is taken as the refractory period of the cardiac phase. Okay, the relative refractory period is the diastolic phase. If supposedly we are giving a shock to the frog on the upstroke area, okay, supposedly I'm giving a shock when the cardiac tissue is on the relaxation period, supposedly this is the point. Okay, so what will happen immediately, because of this extra stimulation, we are going to get a systolic curve. Okay, and that will be depicted with a downward curve. Okay, now after systole, you know the cardiac tissue has to pass through a relaxation phase. Now it will be the relaxation phase because of the extrasystole. Now this point is called the extrasystole. Now this is an abnormal relaxation period. Okay, so it will come. Now the cardiac tissues will be expecting to get a pacemaker signal, but this time around what will happen, coincidentally the cardiac tissue will be found in a refractory period. Okay, because of the previous peak that has been mixed, or previous signal that has been missed. So it will take some time for the cardiac sinus venosus to give or pass the signal to the cardiac tissue. So what will happen, so the time will be elapsed for few milliseconds, and then what happens at this particular time, the volume of the fluid, that is blood in the ventricles, will keep on increasing, and then it encounters another signal. Okay, so this time around, in this point, the normal pacemaker signal comes and it does the normal cardiogram. Now the time period from the relaxation of the extrasystole till it again receives the normal sinus rhythm is called as the compensatory pause. Okay, this compensatory pause is this. Okay, X is showing you the extra systole and this is the compensatory pause for you. Okay, next curve for our discussion is what is the physiological significance of the curve. Now in this curve you can see the curves are showing you two phases which are more or less flattened area. Okay, and the rest of the curves are showing you a typical pattern of cardiac activity. So basically these are, whenever you get a curves like this having some peaks, okay, like this or this, that's a cardiogram for you. Okay, now in this case you see the curves are showing you a normal phase of cardiogram. Then suddenly at this particular point something is happening, there is a pause for some time and then it again starts to regenerate its not cardiac activity. But this time around you can see the cardiac activities are happening at a slower rate, as well as the force of contraction is little bit lesser. Okay, then again at this particular point something is happening, there is some gap in there. Okay, then it again starts to regenerate with a lesser heart rate, as well as the force of contraction decrease. But this time around it does not even look like the normal cardiogram, so that's why it's called as your idioventricular rhythm or asynchronous rhythms. Okay, so this curve, if the question is to identify curve, this curve is showing you effect of spontaneous ligature on frog's heart. Okay, so this curve is showing you the effect of ligature along the conduction pathway of the heart. Okay, supposedly this particular point is called as the first ligature point, and this point is called as the second ligature point. Now where this ligature tied and where, we know the cardiac pacemaker activity of the heart is generated from the sinus venosus, then it passes to the atria. Okay, it gives the conduction to the atria. Okay, generates the conduction pathway to the atria, then the whole atria is contracted. Now then it passes to the ventricles, and from there the conduction pathway goes there, and then it goes to the rest of the vascular tree. Now supposedly you are tying a knot somewhere around here. Okay, what will happen, the conduction or the pacemaker activity is generated in the sinus venosus, but it won't be able to pass to the next chamber. So what will happen, momentarily there will be pause, the atria won't be receiving the signal. Then what it does, it self regenerates. There in the atria also there are some self regenerating pacemaker cells are there, so then they will start regenerating these impulses. Okay, then it will regenerate and pass to the next part of the chamber. But this time around, since this is an abnormal sinus venosus as compared to that of the sinus venosus, the typical sinus venosus, it has a lesser rate of conduction. Okay, so that's why you get a curve having a prolonged RR interval or prolonged period or between two phases of the cardiac cycle. Okay, now supposedly in the next phase you are, along with the first ligature, you are also tying a thread here. This is called as the second ligature, but this is the first and the second ligature. Now at this particular experiment, now what happens, the sinus venosus signals are also not coming to the ventricles, as well as the signals that should have been coming from the atria, that also not coming. So what will happen, momentarily there will be another pause, then the ventricles self regenerates its own pacemaker activity at a much lower rate and force of contraction. So that's why you are getting a curve having a very prolonged phase of the cardiac cycles, and these are called as idioventricular rhythms. Ventricular rhythms. Okay. Now the name stainless ligature, it's the tying of a ligature. Okay, tying a knot with a thread. Stainless is the name of the scientist or the researcher who invented this experiment. Okay, this experiment shows us the effect of electrical conduction pathway. Okay, so how the electric conduction happens. So next experiment, identify the chart displayed the physiological significance of the curve. Now in this curve, the difference between this curve and the previous curve is this. Okay, there is a single plateau phase. In the ligature, I showed you there was a two plateau phase. Now here there is the simple thing to remember in this whole video session. There will be some students who will have some confusion between the stainless ligature chart and a chart which is signifying this. By the way, this is called as effect of vagal stimulation on frog's heart and the phenomenon of vagal escape. Now in the heart of a frog, what happens? The parasympathetic nerves and the sympathetic nerves are both acting together. Okay, but the parasympathetic, it helps in decreasing the heart rate. Okay, so intrinsically what happens, the heart has a if I isolate the parasympathetic nerves and the sympathetic nerves, I'll keep the heart tissue in a chamber with a Ringer's lactate solution, and I tie it to some electrode, then I will be able to see the heart will be beating at a very faster rate. Now this is because of the intrinsic activity of the heart, okay, cardiac tissue. So intrinsic activity of the muscles of the cardiac cells to pump at a higher rate. Okay, it will pump at a rate of 100 to 100 times per minute. Okay, but that is very high. We know our heart usually in a human body, it beats at the rate of 60 to 100. Okay, so why this is lesser than the intrinsic activity of the heart? It is because of this parasympathetic. The parasympathetic comes into play and it decreases this heart rate to a lesser value. Now this is called as the normal vagal tone. Okay, similarly the sympathetics are coming to play only when the stress situation comes into play in your life. Okay, it increases the heart rate. Now the normally in day-to-day life, when you are doing a day-to-day work, the vagal tone is every time, in every second of your life, is acting on your heart. This is called the vagal tone of your body. Now supposedly, if you are stimulating the heart along with the parasympathetic, now you are stimulating with a higher magnitude of current. What will happen? The vagal nerve will first of all decrease the rate of signal transmission in the heart. And what will happen in this chart, for the first few minutes, if supposedly I am using a lesser amplitude of signal in the vagal stimulation, for the first few seconds, first few curves, you will get a lesser amplitude of curves. Okay, so supposedly you will get a lesser amplitude of signals. Okay, now if supposedly you're increasing the stimulation, the upper signal curve is showing you the display chart of the point from where to where you are giving the vagal nerve stimulation. If supposedly is a lesser magnitude signal, then you will be able to get some curves having lesser amplitude and a slower force of contraction, or it will be spaced It will be spaced out and there will be some curves. If supposedly with you're giving a higher the maximum magnitude of current that you can possibly give with the instrument on the experimental setup, what will happen in that particular time? So this is the point at which you are maximally stimulating the vagal nerve. What will happen? The heart will momentarily throughout the point will get inhibited in its cardiac activity. So that is called as your cardiac inhibition. But as I told you, it is also supplied by the sympathetic nerves. Okay, along with that, okay, the parasympathetic nerves, it requires the help of acetylcholine neurotransmitters. Now these acetylcholine neurotransmitters are present in the vesicles of the nerves. It has some limitations in supply. There will be a time when the supply will get depleted by the destruction of the acetylcholine by the cholinesterases. So what will happen? This acetylcholine will be depleted. Okay, along with that you will also get the so by parasympathetic it is actually inhibiting the pacemaker cells. But I've told you, when the pacemaker cells are inhibited, what happens? The next line of pacemaker cell, that is the atria or the ventricles, will come into play. So a time will come when you will get a idioventricular rhythm because of the next pacemaker cells will come into play. Okay, second physiological reasoning of this curve is depletion of acetylcholine transmitter because of the destruction of the acetylcholine and the limitation of the source in the vesicles of the nerves. Third is the sympathetic come into play, that is sympathetic stimulation, okay, or overdrive. Okay, so the sympathetic will take the upper hand, and from this point you will again tend to get slower by slower you will try to get a cardiogram. Okay, so that's called as your effect of vagal stimulation on frog's heart and the effect of vagal escape. Now this point at which you are starting to get a cardiogram, that is called as the point of vagal escape. Okay, the vagal stimulation is escaped at this point. Now next question might come, what is the effect of the drugs, or the identify the chart displayed, what is happening in the part mentioned X. Okay, so these are the effect of drugs. Okay, effect of drugs or ions in frog's heart. Okay, so instead of this word drug and ions, you can, in this particular case, you should add the word calcium. Effect of calcium ions in frog's heart. Okay, if suppose you're given a curve in which identify this, they erase this area and they wrote X over here. Okay, then what would be the answer? Acetylcholine. In this case, if they erase over here and they give X over here, that means what is acetylcholine. Okay, why it is happening? Now adrenaline as such is a sympathetic drug. Okay, sympathetic group of drugs. So and acetylcholine is a parasympathetic drug. Okay, now the mechanism of action of sympathetic drug is that it will help in increasing the rate and force of contraction of the heart cardiac tissue. So it will affect the cardiac pacemaker cell, it will affect the myocardial cells. So what will happen? You will tend to get increasing amplitude and the rate. The curves will be coming closely spaced together. Okay, clumping together. Whereas in case of the parasympathetic drug, work happens, so as soon as it is supplied, okay, so the picture is not accurate. Okay, so as soon as you start to apply, what happens, the curve will show a lesser amplitude, as well as will have a, so heart rate will decrease and the force of contraction will also decrease. So you are getting this kind of a curve. Whereas in case of the calcium, what happens, if you are supplying calcium has an effect of calcium rigor. So as soon as you start to give or supply calcium, what happens, the cardiac tissue starts to form a, so cardiac tissue with the calcium chloride as such will help in increasing the force of contraction. Okay, and it will decrease the relaxation time. Okay, so it won't get an ample amount of time for relaxing. So as a result of what happens, there will be decrease in the end diastolic volume, and what will happen, in spite of the increased force of contraction, what happens, there is lesser amount of the blood, so amplitude keeps on decreasing. A time comes, the heart will remain in a statically contracted position, that is called as the calcium rigor. So it will stop in a systole. The heart stops in systolic phase of the heart. Okay, whereas when you supply the potassium, or potassium in the form of potassium chloride, what happens, so potassium as such will decrease the resting membrane potential of the cardiac tissues. Now when this is happening, what happens, the tissues become less excitable or inexcitable. Okay, so it will, so when the cardiac tissues are not responding well enough, so that means what, it's not in a systolic phase, it's in a diastolic phase. Okay, it's in a it's a partial relaxation. So at time will come, the rate will decrease. Okay, the force of contraction will decrease and the heart will stop in a, so this is the point is which is called heart stops in diastole. Okay, when the heart is stopping at a higher level than the baseline, it's called as the calcium rigor. When the heart cardiac tissues or cardiogram is stopping at the baseline, you would think that to be the potassium rigor. Okay, or the effect of the potassium. So next question or charts or spots are, identify the curve, what is A. Okay, so identify, next question, this question can also be framed as, identify the part mentioned X in the chart displayed below. What, write the causes of the phenomenon being displayed over here, or simply they may give a pictorial depiction of the chart and ask you about the physiological significance of the card. So all the three curves are showing you the phenomenon of genesis of fatigue. Okay, so as such, in definition wise, fatigue is defined as the decrease in performance due to continuous and prolonged activity. Okay, so if you continuously using the nerve muscle preparation with a shock and recording the simple muscle twitch, okay, so you are getting multiple sets of simple muscle twitch. Okay, you are rotating the kymogram and repeatedly stimulating at the same point. Okay, so you are getting a curve chart having a simple muscle clumping of the simple muscle curve. Okay, so plenty of, at the same point you are stimulating, you are getting plenty of curve. But the interesting point to note down over here is that for the first set, so this is the first set which is showing you the curves to be like this. Okay, then the first few second sets of curve are having a higher amplitude, then the third set of curve arrived with a lesser amplitude. Okay, and having more duration. Okay, lesser amplitude and more latent period. Okay, so simple way, let's look into this curve more clearly, you will understand. So what will happen, So this is the first point of stimulation, okay, and this is the latent period. So this is the first curve that you are getting, okay, first curve. Then due to the beneficial effect of the first curve, as explained in the first few charts, the latency and as well as the amplitude will increase. Amplitude will increase, but the latency will decrease because of the beneficial effect of the previous curves. A time will come, what happens, there will be some detrimental effects that will be accumulated in the experimental setup, okay, or in the muscle tissues or in the nerve tissues, okay, whatever in the nerve muscle preparation, there will be some detrimental effect, and because of that, what happens, gradually the next contractions that will be coming on next curves that we were getting are having a lesser amplitude and a longer latent period. A time will come, you will see that a curve that is arriving which is not even touching the baseline, it is going through and through. And I've told you, a simple muscle twitch, if it has to pass through a relaxation, it has to touch the baseline. That means it is not touching the baseline, that means it is a muscle which is in a continually contracted phase, okay. So this is called as contraction remainder. That means contraction is remained. So this A and X is showing you the contraction remainder. So by the way, this phenomenon that is achieved, there is a fatigue, is a reversible phenomenon, and there is no permanent damage in the nerve muscle preparation that we are doing this experiment, okay. So physiological significance of this experiment is fatigue is achieved because of the depletion of the factors which need it to show the simple muscle twitch. Now for a simple muscle twitch, what has to be there? First criteria is there should be the neurotransmitters, okay, in this case the acetylcholine, okay. The second factor is it also should need nutrients, okay, oxygen, okay. The third factor is there should be less amount of metabolites, okay, that is the waste products after a cell does the activity or work it does. Every cell in our body, after doing work, it emits some metabolites which has to be taken out from the tissues in order to work properly, okay. So these are the factors which needs to be present, okay. So if these factors are not satisfied, then we will get a time when the muscle cease to do work, okay. That at that particular point it achieves the fatigue point, okay. So the fatigue is achieved where? So I told you we are doing this experiment on a nerve muscle preparation. A nerve muscle preparation is made of gastrocnemius muscle or the calf muscle with the sciatic nerve, okay, and we are conducting the experiment by giving a shock to one part of the nerve. The signal transmits through the nerve, okay, it contracts the muscle, okay, and the muscle pulls the thread and the lever and it depicts the simple muscle curve. If supposedly there is fatigue achieved, there is a contraction remainder curve is achieved, that means the fatigue point has achieved. What does this signify? There is a problem somewhere around here in the nerve or in the muscle or in the new musculation. There are the 3 points where the problems will be there. Now immediately when the muscle has achieved fatigue, okay, if supposedly you are directly stimulating the muscle, okay, you again get a simple muscle twitch, okay. On the other hand, if you are directly stimulating, so this was the curve when we are getting stimulating the muscle directly. If supposedly we are stimulating the nerve directly, then also we are able to get a simple muscle twitch. and a nerve is practically unfatigable. Okay, always it is regenerating the signals. It's a electrical conducting pathway only. Okay, so what could be the problem? So the problem could be this junction there is the neuromuscular junction. So our fatigue is generated in the neuromuscular junction, and at this particular point only the acetylcholine is depleted and all the nutrients are depleted. Okay, this is depleted acetylcholine, nutrients and oxygen depleted, and the metabolites are increased, and because of that fatigue is happening. So this is the physiological significance of the curve that is being displayed.